everyone. Welcome back to the WebEVTalk series. It's on the 8th November in the USA, but 9th November in Australia. So today we have an early career scientist, Abhimanyu Thakur. He's from University of Chicago in the USA. So Abhi research engaged multidisciplinary approaches focusing on exosome bioengineering, cancer research, biosensing, theranostic drug delivery, and neurodegenerative diseases. He received PhD in biomedical sciences from the City University of Hong Kong, CTUHK. So continuing as a postdoc in the Center of Regenerative Medicine and Health in Hong Kong. Currently, he's working in the University of Chicago in the USA. So he has multiple awards, including a Young Investigator Award by American Association for Cancer Research, Korean Cancer Association, Global Young Scientist Summit Award by Nanyang Technological University, Singapore, the Choi Ye-ching Research Award, and Outstanding Academic Performance Award by the CTUHK. So his talk today is about exosomal MCT1 and CD147, role as biomarker and functional mediator in glioma progression. So before we start, I'd like to thank Horiba Scientific, who's been supporting WebEVTalk since May. Thank you so much for supporting this session as well. Go ahead.
Is it fine now?
It's good now. Go ahead,
Okay.
please.
So hi everyone, I'm Abhimanyu Thakur, and currently I'm a postdoc in University of Chicago, Pritzker School of Molecular Engineering. And today I'm going to talk about this exosomal MCT1 and CD147 as a role in its role as a biomarker and functional mediator in glioma progression. So this work was carried out at City University of Hong Kong. It was a part of my PhD work. So what is glioma? So glioma is a brain cancer which occurs in central nervous system that is originated from various neuroglia stem cells, and its occurrence is around 1.7 million patients as per 2017 statistics. And the most aggressive form of glioma is the glioblastoma. So even though there are treatments available like temozolomide in terms of chemotherapy or even radiation therapy and surgery, but the average survival rate for a glioblastoma suffering patient is only 14.6 months. So that shows how crucial it is to study this glioblastoma. Well, in most of the cancer cells, one of the important hallmark of cancer is the metabolic reprogramming, and in which there is a high glycolysis and there is a high formation, increased formation of lactate in intracellular area of the cancer cells. And to take out these lactate in the hypoxic tumor microenvironment, there are two important glycolytic proteins like MCT1 and CD147, which overexpress on the membrane of the cancer cells, and they help in the exocytosis of this intracellular lactate, and thereby they form a metabolic coupling with its stromal cells, and together they contribute to the tumor progression. Therefore, this metabolic reprogramming is very crucial for survival of cancer cells. So what is this MCT1 and CD147? So this MCT1 is a lactate transporter which work together with its binding partner, that is CD147, and they play important role in migration, proliferation, and angiogenesis. These are the important tumor phenotypes. And it has been found that their inhibitors act as anti-glioma agents, and they have been found to be crucial indicator of glioma malignancy. Although we know that there is enhanced expression of MCT1 and CD147 in glioma, but and also there is a dramatic release of exosomes from glioma cells, but we don't know how this increased expression of MCT1 and CD147 control the enhanced release of exosomes from glioma. Well, what are these exosomes? These are small extracellular vesicles which are around 30 to 200 nanometer, in diameters, and they are released from most of the cell types and even biofluids like blood, cerebrospinal fluid. And they play important role in cell intracellular communication, even metastasis. And there are certain mutant proteins which are specifically found in glioma, like EGFRV3, IDH1, and there are certain microRNAs which are specifically found in glioma cells, like microRNA 21. It has been found that these exosomes can cross the blood-brain barrier and even the blood CSF barrier, and they are found to be present in the peripheral circulation. Interestingly, these exosomes can mimic their parent cells from which they have been derived. Therefore, these exosomes can be an important platform for the detection of biomarker to track the malignant glioma progression. So although there are various conventional methods for the detection of the cargos, for the detection of this glioma, like clinically MRI or even CT scan are available. And in terms of biochemical methods like electron microscopy or ELISA or Western blots, this can be used for the detection of the cargos, like especially like proteins in the exosomes. However, they suffer with certain limitations as they cannot detect early. And these biochemical methods are either time consuming, or they are indirect method, or they have like limited sensitivity, or there is a chance for protein contamination, and there is a requirement for the large volume of samples. Therefore, the detection of precise molecular signature of glioma progression has been challenged. In that scenario, we propose to use LSPR and AFM based biosensing system, which are non-invasive and can be used for precise monitoring, and these are label-free methods. So these biosensors can be used for the detection of exosomal biomarker. So what is this LSPR? LSPR is surface plasmon resonance. It's a technique for detecting changes in refractive index at the surface of sensor due to antigen-antibody interaction or even protein-protein interaction. And they are, importantly, these are label-free, and they can detect the exosomal cargo with high sensitivity. So in this LSPR system, there is a sensing chip on which the antibody against those antigens, they are immobilized by using various functionalization method. So in this study, we used gold nano island, self-assembled monolayer gold nano island LSPR. And what happens is whenever there is an interaction of the protein or the antigen with the antibody, there is a rise in this association curve. And whenever there is a detachment of those antigen from the antibody, there is a dissociation, which is followed by this regeneration curve. So this was used for the detection of exosomal cargo. Second method, we use this atomic force microscopy. It's a very high resolution type of scanning probe microscopy, which can even detect with a resolution of like fractions of a nanometer can be detected. So this involves AFM cantilever and a sample disk. This AFM cantilever, they contains this AFM tip, and this AFM tip is used for probing those samples on the AFM sample disk. So traditionally, scientists have used mica surface or even gold over the mica or glass surface. Here we used gold nano island sample disk for better resolution, and it has a typical AFM force curve. Whenever there is whenever there is an interaction of the antigen and antibody, this force adhesion force curve we can obtain. So one of our objective of this study was to detect MCT1 and CD147 in glioma-derived exosomes by level-free LSPR and AFM biosensors to track glioma progression. So our first question was whether these hypoxic glioma cells contain enhanced expression of MCT1 and CD147 as compared to the normoxic glioma cells. So we did RT-qPCR, and even we did immunocytochemistry and Western blot, and we found that under hypoxic condition, there is an enhanced expression of this glycolytic genes like hexokinase or LDH, and together with this our target proteins MCT1 as well as CD147. which we further validated using this Western blot. And we also checked its distribution pattern in the glioma cells under hypoxia compared to the normoxia. Here we can see that there is an enhanced expression of MCT1 as well as CD147 compared to the normoxic glioma cells. So our second question was whether this MCT1 and CD147 are crucial for malignant glioma progression. Although many scientists have already shown like how this MCT1 and CD147 play important role in many different cancer cells, including glioma cells. So we again did, we analyzed the migration phenotype as well as proliferation phenotype. And we found that under hypoxic condition, there is an enhanced migration compared to the, compared to the normoxic glioma cells. Similar pattern was obtained in the proliferation assay. To further understand whether this is indeed due to the MCT1 and CD147, we studied gain and loss of function, and we did overexpression as well as knockdown of MCT1 and CD147. So we again found that upon overexpression, there is an enhanced proliferation as well as migration. Whereas when we did knockdown, there is a inhibition of this proliferation as well as this migration. Next, we questioned whether this MCT1 and CD147 control the enhanced release of exosomes from malignant glioma cells. So we analyzed the release of exosomes under normoxia and hypoxia. We found that under hypoxia there is a significant increase of exosome release, which was again found that upon overexpression of MCT1 as well as CD147, there is an enhanced release of exosome. And upon knockdown, there is a reduction in the exosome release from glioma cells. This shows that there is a connection of this MCT1 and CD147 in the exosome release from glioma cells. So what is the probable mechanism behind the enhanced release of exosomes from this malignant glioma cells via this MCT1 and CD147? So we analyzed the intracellular calcium, and we found that upon under hypoxia, there is an enhanced expression of this enhanced calcium level, and which was inhibited by using this BAPTA-M inhibitor for calcium. And we found that under hypoxia, this is enhanced, and which was further found that upon overexpression of MCT1 as well as CD147, there is a enhanced release of exosome, which was further reduced by using this BAPTA-M, that is calcium inhibitor. So this shows that there is a certain connection of this calcium through this MCT1 and CD147, because when we are inhibiting the calcium after the overexpression of MCT1 or CD147, there is a decrease under this overexpressed condition of the exosome release. Further, to understand what's the connection of the tumor microenvironment related lactate. So when we treated our glioma cells with the sodium L-lactate as a representative chemical for the lactate tumor microenvironment, we found that there is an enhanced calcium level, which was shown by Fura4AM level glioma cells. And upon treatment with 20 micromolar BAPTA-M, it was decreased. Similarly, we found that under this lactate treatment, when there is a inhibition of this MCT1 and CD147, we found that there is a release of this, there is a decrease of this exosome release. So this shows that the glioma cells showed intracellular calcium dependent release of exosome under hypoxia as well as lactate environment. Next, we questioned whether this MCT1 and CD147 are present in glioma cells derived exosomes. For that, we analyzed by using various different methods like immunogold electron microscopy, and even Western blot and ELISA. We found that there is an enhanced expression of MCT1 and CD147 under hypoxic condition by immunogold EM as well as by Western blot and even by ELISA. Next, we question, can this LSPR and AFM precisely detect the MCT1 and CD147 in glioma cells derived exosomes? So for that, we used the strategy of this LSPR. So what we did is we functionalized our SAM gold nano island LSPR using the antibody against MCT1 as well as CD147. And in the AFM, we immobilized our exosomes by using CD63, which is one of the marker of the exosomes. And we probed for the MCT1 by functionalizing the AFM tip with MCT1 antibody. And here in a separate experiment, we did similarly with the CD147. So interestingly, what we found is there is an enhanced phase response under hypoxic condition, both for MCT1 as well as CD147. Then we examined the topography. This is a 3D diagram, 3D figure of a single exosome, and this is the height curve, which confirms the size. And again, using the AFM, we found that there is an enhanced force curve compared in the hypoxic glioma cells for MCT1 as well as CD147 compared to the normoxia. Next, we question whether the level of this MCT1 and CD147 is correlated in parent glioma cells and their derived exosomes. So for that, we did overexpression as well as knockdown of MCT1 and CD147. So to our surprise, we found that when we did overexpression of MCT1, we found it is correlated with this enhanced phase response as well as enhanced AFM force curve. And we found that it is correlated from its parent glioma cells as well as this daughter exosomes. Next, we questioned, can this increased level of MCT1 and CD147 be detected in exosomes derived from blood serum of mouse model of glioma by label-free this LSPR and AFM biosensors? For that, we established xenograft mouse model by injecting UAT7 cells in the SCID mice, and we examined the tumor development by using MRI, and we isolated exosomes from the tail wheel, and we found that there is an enhanced expression of this MCT1 as well as CD147 in both U251 glioma cells as well as UAT7 glioma cells derived based xenograft mouse model. Similar pattern, we found the enhanced force curve in U251 as well as UAT7 xenograft mouse model compared to the wild type. Again, we found that there is a correlation of the biophysical properties of glioma cells derived exosomes, and which can be used as an additional biomarker for malignant glioma cells. So what we did is we analyzed the zeta potential under the normoxic and hypoxic glioma cells derived exosomes, and then we examined various biophysical properties like roughness, Young's modulus, which is a measure of stiffness, and also the adhesion force. And we, for the first time, saw that there is a correlation of this MCT1 and CD147 with the biophysical properties of the glioma cells, which was further correlated with the enhanced tube formation in the endothelial cells after the uptake of this hypoxic glioma cells compared to this normoxic glioma cells. So overall, we found that there is an enhanced intracellular lactate in tumor microenvironment, and to take out this lactate out of the cells, this MCT1 is overexpressed on the membrane together with its binding partner CD147, and it play an important role through this calcium pathway for the release of exosomes, which can be further taken up by the endothelial cells and enhances the tube formation. And these released exosomes further further mimics its parent cells in terms of MCT1 and CD147 expression, which can be detected by this level-free LSPR and AFM biosensor. So this work was published in Science Advance, and we got highlight in various news news agencies. And this is my boss. And I finally acknowledge all the group member who helped in carrying out these experiments, and our collaborators and all our funding agencies. Yeah, thank you.
Thank you so much, Abhi. Wonderful presentation. I wonder if anyone wants to start asking questions. Please go ahead. Please, Michael, go ahead.
Okay, Phil, I think your guitar amplifier is up too loud. Anyway, I mean, that was really nice. One thing that this is really reminiscent of is what they call the unfolded protein response. It causes this release of calcium into the cytosol, which we think also then causes extracellular vesicles to get released more actively. And when this is induced in cells, the MCT1 and CD147 do tend to go up. Did you actually take a look at that? Because hypoxia is actually a very potent inducer of this thing called the unfolded protein response.
So do you mean, did we check the expression of MCT1 and CD147 in the exosome?
Well, what I was wondering is, I don't know how hard you looked at the cells as opposed to the exosomes, but there would have been indications of what's called the unfolded protein response in the cells. They would have increased expression of certain chaperones like the HSPB1 and which is GRP94, GRP78, things like that. Because everything you showed looks exactly like what happens when the unfolded protein response is induced in a cell line. And hypoxia is a very known potent inducer of that. So if you didn't do it, I wouldn't be surprised, because who would look for this except some clown like me? But the idea being that that might be another mechanism to look into check these particular proteins in there, because this really looks exactly like an unfolded protein response outcome.
Yeah, I think certainly there might be other mechanism. But we just explored one of the mechanism, that is through the calcium pathway. Yeah, but actually what we found is we have shown by using various multiple methods the expression of this MCT1 and CD147 in exosomes. So I think that makes clear that there is a enhanced expression of this MCT1 and CD147.
No, right. And so what happens when you induce this unfolded protein response is it causes those two proteins, among others, to get increased expression, especially in cancer cells. And in that case, that calcium release into the cytosol is actually very typical of that response as well. So I'm just suggesting that if you want to carry out your next paper, start asking questions about the unfolded protein response, and that being sort of a built-in mechanism for this enhanced expression of these two molecules, and putting them on vesicles. Now that's a different area. I don't know how that's sorted out like that. That's going to be a cool thing to dig into. But we've noticed, and other people have noticed, that when you stress cells in this way, they make more vesicles in general. I haven't actually looked to see what are the differences in those vesicles, but this looks like a good place to start. But I was just struck by how similar this looks to things that we see when we refer to it as the unfolded protein response. The calcium efflux is from calcium channels that open from the ER, allowing it into the cytosol. And this is putting all the rest of that together into this sort of thing as this biomarker was, I think, a really cool way to look at these things. The other question I had is, how long does it take to do these analyses? Say you're a clinical lab, somebody rushes to you a blood sample. How fast can you get through this with either the atomic force microscopy or the surface plasmon resonance?
Oh, I think if everything if everything is optimized, it will only take 10 minutes something. Yeah.
Wow.
10,
okay,
minutes.
So that's a real clinical assay then.
Yeah, the optimization is very important. So it took a lot of time to optimize, but once it was set up, yeah, we repeated several times. Yeah. Yes.
Okay, that's pretty cool.
We are first confirming the 3D image by scanning, and there is a point and shoot application in that software. So we are shooting at various different like 100 or 200, and we can simply take what's the force curve like this. Yeah.
Nice. That's really cool. I'll let Phil's got a bunch of questions. I'll let him or Carolina can read them over.
Actually, Michael, I was wondering, have you actually come across an unfolded protein in the EVs? Can you tell actually?
That's really hard to do. I've recently learned of a potential way to do this, and I don't know exactly how it is. I'd have to work with somebody at like Stanford, because you know how bad they are. But the idea, we don't have, like when you have for the SOD1, which aggregates, there are really cool antibodies for that. For your average sort of unfolded proteins, we don't know that. And the other thing is, every time you handle, say you try to lyse open a vesicle, how sure are you didn't cause the unfolding yourself?
That's such a good point. That's true.
So, but this is something I've really been interested in, and we just don't have to my knowledge, there's no good model for this, no good way to assess that. That's not going to have some built-in problems.
Yeah.
I see.
Okay, that's cool.
Yeah.
Okay, Phil's questions. Of the increase produced exosomes, what percentage are positive for the markers you follow? And from a true glioma not aligned, what about PDL1 and CTLA4? This is probably two questions.
Yeah, so the first question is what percentage of exosomes are positive for markers?
I think with the AFM, you can actually tell which is positive from the marker or not. That's true.
Yeah, I think what we found is, I think it's around 80% something. Yeah, 80 to 85% something for the MCT1 as well as CD147. And whether it is from true glioma not aligned. Yeah, actually one of the reviewer, they asked to perform in the true glioma, and we did experiment in the supplementary. Yeah, it's there, and we examined the MCT1 as well as CD147 similarly in the true glioma-derived exosomes. That is a stem cell, patient-derived glioma stem cell. Yeah, so that was one of our critical experiment. The reviewer asked, yes. And what about PDL1 and CTLA4? So PDL1, yeah, this is one of our upcoming project, still it's ongoing in our lab, and one of our PhD student is doing. I'm not currently in that lab now. Yeah, and CTLA4 we did not examine. Yeah, PDL1, yeah, of course, it's ongoing in my previous lab. And in patient plasma, what percent of total have markers? Okay, so this MCT1 and CD147, for a long time it has been, even it has gone to clinical trial even, this MCT1. Yeah, so like AZD3965, this is one of the inhibitor of MCT1. So as you are asking, like what percent of plasma? Yeah, I did not check this one. Yeah, but I know in tissue level they have shown. Yeah, but I haven't checked for the patient plasma.
So Abhi, are they actually localized at the early endosome in the cells?
No, I think they are they are mainly on the side plasma membrane. Yeah.
Okay, all right. Because current understanding about the definition, but exosome is those that coming from intracellularly. So there's a potential that's plasma membrane derived, like small extracellular vesicles rather than the exosomes.
So I think there might be a chance, like while following the what is that lysosomal endosomal pathway, these exosomes, when they are interacting with this plasma membrane to ooze out of the cells through this plasma membrane, they are taking those proteins, and that's how they are mimicking. Yeah.
How do you isolate your vesicles, just for the audience to know?
Oh, so we actually use two different methods for For the larger experiment like Western blot, this kind of experiment, we used differential ultracentrifugation. But for wherever there is a requirement of very small volume, in that case, we used Thermo Fisher reagent. Yeah. Like especially from blood, we use Thermo Fisher commercially available reagent.
Okay. All right.
So let me throw out another question here. At University, or you're at University of Chicago now, right?
Yeah.
Do you have access to patient blood, patient plasma?
Yeah, here we have, yeah. Here.
Okay, I was going to say we have a pretty extensive bank, and if you wanted to have some of that to play with, we'd be glad to let you try it.
Oh, okay, that will be great, I think. So currently, I'm looking for Alzheimer patients, yeah, blood sample.
Okay, I can put you in contact with people for that.
Okay.
So
yeah, maybe I'll contact you.
Yeah, yeah, we have a pretty active Alzheimer's group that's actually involved in vesicle work, so they could be interested in this.
Okay, yeah, that will be great, I think. Yeah.
Okay. If there's any more questions, otherwise I'll close the session even though it's a bit earlier. No more? Okay.
Yeah.
All right. With that, and then I'll conclude the session. So next week, sorry. Apologies. Abhi, thank you so much for presenting and a wonderful study that you've shown us today, and a lot of models that you use, and interesting development as well for the detection, use of biomarker for the glioma, and good luck for your next study with the other neurodegenerative diseases. Thanks so much for sharing with us.
Yeah, thanks for providing this opportunity. Yeah.